5. It's an obligate wetland species. I've sorted so that it progressively goes from a wetness index of -5 to -3 and so forth until we get into the positive numbers, the +5. So here are the more upland dry species. So it's already sorted, and I imagine if you're doing some sort of wetland delineation or something like that, species, of course, that are hovering around 0 are of particular interest, right? They're sort of, oops, did not do that right. They're sort of neatly arranged for you, roughly right here. I imagine those species are of particular interest. Then I have a family key in which all of the families of fern-like plants are arranged. And this is, again, right out of Michigan Flora. And in fact, there are hot links so that if you're out in the field and you do have Wi-Fi connections, you could just tap on this and it will take you straight to the Equisetaceae, for example. And then the same thing for lycopods. So if you've, through the family key, identified that you're dealing with some kind of a lycopod, then when you go to the lycopod key, you can at least take it to genera. And of course, if you can activate the hot links, then you can go in closer, go link to Michigan Flora and key out the species. But at least I get you to genus, if not sometimes species. Same thing for the Ophioglossaceae, whatever they are, I'll describe them in a bit, and then the Dryopterids. So there's, in a sense, 3 more specified keys: the lycopod, the Ophioglossaceae, and the Dryopterid. It's not all of the families, but these 3 keys are going to get you the vast majority of the fern species that you might encounter in Michigan. So I'm going to leave it at that, except for to say that this first spreadsheet here, this last column here, notes, are notes that I've put in, just things I happen to know about the species, or notes that I thought were particularly insightful from Michigan Flora. I hope to kind of keep filling that in the future. But I made sure to make notes about species that are particularly difficult or particularly important within the flora. And like I said, I'll try to continue working on filling that in over time. So I'm going to stop sharing that. That's all I have to say about the spreadsheet, unless you have a question about it. So I'll sort of continue on. So the spreadsheet, here's what's in it. It turns out exactly 100 species recognized in the Michigan flora that we could call fern or fern-like plants. I'm always taken aback when the number is so rounded out like that to 100. And then within that family key, that gets you to about 21 different families. And then, like I said, there's a Lycopod, Ophioglossaceae, and Dryopterid key that really is going to be useful for the vast majority of ferns and fern allies that you might encounter. So I thought I'd begin with a little bit of sort of large-scale taxonomy. Kind of helps you, at least helps me mentally, kind of nest these different plants because they are distinctive from one another. And so this is a phylogram that I constructed for a class I teach called plant morphology. It's nice and simplified, so just major branches are there, and students can kind of crisply see relationships between major clades of plants. And so just to give you a quick tour, of course, this is the plant kingdom. Bryophytes sort of split very early in the evolutionary history of plants from the tracheophytes. The tracheophytes are just plants that produce lignin. It's not true that they are the vascular plants. There are bryophytes, as you probably know, that have vascular tissue. So bryophytes have the unfortunate misnomer of being called the non-vascular plants. It's not true. But what they don't have is lignin. And of course, lignin is that key compound within xylem and sclerenchyma, if you know your plant tissues, that imparts hardness, a stiffness to plant parts. It's how trees can stand up. So the trachophytes are much larger. And there's then an early split during the early Devonian into basically two big clades, the Lycophyte clade and the Euphylophyte clade. A lot of big, ugly words. But the key thing about these two clades, the Lycophytes and the Euphylophytes, is that they separately evolved roots and they separately evolved leaves. Or maybe I should say that the mechanism by which they evolved leaves was different. So it looks like the Lycophytes evolved leaves only once, but distinctively through a distinct process than the Euphylophytes. It turns out that what it means to be a leaf has evolved numerous separate times within the Euphylophytes, but each time more or less in the same manner. That's why they can sort of be recognized. It's one of the reasons they can be recognized as a distinct clade. Certainly, if nothing else, they evolved roots through a common but distinct process from the Lycophytes. So that's the big split. And then within the Euphylophytes, some plants release spores, just like bryophytes and lycopods do. Some plants do not release their spores. They make spores, but they don't release them. Instead, what they release are seeds, right? So the seed plants. So what I'm going to cover with you folks today is this paraphyletic group, just meaning a grouping that does not share a common ancestor that is unique to that grouping. So we have seedless plants. They don't make seeds. They release spores, free sporing plants. They're lignified, but they have separate origins. And so these are often, as a result, called pteridophytes, meaning literally winged plants, which is a strange name. The lycophytes are often called the lycopods. And then this big clade over here are typically called the ferns, even though it would include things like horsetails right here. A horsetail is technically a fern because it does fall within this manilophyte clade. So gives you a little kind of big scale tour of what I'm going to talk about here, this grouping here. Lignified, but release their spores, they don't make seeds. And then I think a bit of context about how Michigan sort of ranks with the rest of the world in terms of the diversity of these types of plants. I suppose the two or three punchlines here. Within these pteridophytes, these seedless vascular plants, the Isoëtaceae do not occur at all in Michigan. In fact, they're strictly a tropical and subtropical group. And that's also true for the Pteridophyte, which are also strictly tropical and subtropical. Both groups are found equatorially throughout the world, but are not found in temperate zones. So they're not in Michigan. We have, as I said, about 100 species altogether of pteridophytes in Michigan out of, and this is an underestimate, but I get this from ITIS, the International Taxonomic Information System, estimates that the number of species of pteridophytes in the world is about 12,500. Other sources that I have that I trust a bit more, like the Smithsonian, put it closer to about 15,000. Nevertheless, Michigan contains less than 1% of the global diversity. But given Michigan's size and location, that's pretty impressive. If we were in Arizona, it'd be much lower. And of course, compared to seed plants, Michigan, interestingly, has a much smaller ratio of the seed plant diversity. So Michigan in some ways has a strong representation of pteridophytes. So back to this phylogram, I'm just want to alert you to where I'm headed next. I'm going to talk about these lycopods. Again, they've evolved both roots and leaves through very unique processes from other plants. And as you can see, there's three families within the lycopods: the Lycopodiaceae, the Selaginellaceae, and the Isoëtaceae, and all three are here in Michigan. So that's the group I'm going to look at first. I should pause and see if you have any questions that I could answer about maybe the phylogram or the spreadsheet. I always, what is it? It's Friday, almost 4 o'clock after, I guess, a short work week, but you know, I always hate just talking at people. So please do feel free to interject. I'm kind of hoping you will, because I know what it's like to be talked at. Every now and then I sit in on a lecture or go to a conference and sit in on presentations, and I'll be sitting there drifting off in my mind thinking, how are my students doing this? So I hope I don't lose you here. So the lycopods, the Lycopodiaceae is the first group, and a few things to know about them as a family. The Lycopodiaceae tend to have pretty small leaves, usually less than a centimeter long. However, I can tell you down in the tropics, their leaves can be considerably longer than that, good 2 or even 3 centimeters long. The leaves are helically arranged, which I suppose is maybe most apparent in this photo here. They're homosporous, just meaning every spore is the same in size. And the resulting gametophyte, if you remember your plant biology, they're all potentially at least hermaphroditic, capable of producing both sperm and egg. And so homosporous plants tend to be fantastic colonizers of remote locations because a single spore can colonize that location. That's not true for heterosporous plants that make small spores that develop into male gametophytes and large spores that develop into female gametophytes. Then you've got to have one of each and within proximity of one another to exchange sperm and egg. So heterosporous plants aren't such great colonizers. These are, because one spore will do, and then it can self-fertilize and establish a population. Within Michigan, all of the genera in the Lycopodiaceae produce a cone, a strobolus, except for Huperzia. And that's what I have pictured here is the exception. This is Huperzia lucidula. This particular specimen is from the Leelanau Peninsula. And this particular genus is also unique in Michigan in that it makes gemmy. So first, a couple of things I wanted to show you here is that in these leaves here, hopefully you can make out that there's some kind of little bean-shaped thing on the adaxial, that is the upper side of each leaf, at least within this zone. And those yellow discs or beans you see in there are these sporangia here that you see in this picture in the lower left-hand side. I stained it with toluidine blue, that's why it looks green. It was yellow. I found that when I stained it, it photographed better. You could see the sporangium more distinctly from the leaf. So the sporangium sits on top of the leaf. It's sort of a bean or kidney-shaped thing, and releases spores that are all the same size. This leaf attached down here, so here's where the leaf was torn off of the stem, just to help orient you. Huperzia can also reproduce through gemmy, that is asexually. And here you're looking down on one of these shoots. You can see a gemma right here. This is one, has sort of it's a 3-parted affair. You can see another one here. There's another one right here, and last another one there. You can see them laterally near the tip of the picture on the right. So the gemma, of course, can just break off, tumble around, and then they will root and establish asexually a new plant. The spores, of course, can fly for any amount of distance and colonize locations more remotely. So that's Huperzia. It's the only genus, it's the only species, really the only genus, I should say, within the Lycopodiaceae that does not make a cone, but instead makes gemmy. Here is a different species, still in the Lycopodiaceae. This is Spinulum anotinum, and you can see it clearly makes cones. Again, I'm going to jump back to the prior slide. The sporangia not arranged into a cone. It kept growing, right? So it was vegetative, then fertile, then vegetative. Whereas this species produced cones. More about this species in a minute, but it's easy to potentially confuse some of these species. One thing to also look for is that Huperzia makes these interesting annual constrictions, meaning come fall, when it ceases growing, the diameter of the stem actually shrinks a tiny bit. And then, of course, when it resumes growth, it has to start at that constricted point and then expand out. So you can sort of see right here, the shoot gets narrower, and right here too. So it was wider, got narrow, and then it's widening out again. That's unique, again, to Huperzia. None of the other members of the Lycopodiaceae do this. They have cones, they don't make gemmy, and they don't have those constrictions in the stem. I'm going to jump back up again and say, obviously, Huperzia is very widely distributed throughout the state. I'm rather surprised that some of the counties shown don't have records for it. I suspect it's there. Very, very common. Spinulum's pretty common, but more in the sort of northern half of the state. And especially Spinulum, but it's true for Huperzia too. They can form these dense colonies that can dominate patches within wetland or forest. I was once astonished at the acreage, literally the acreage that was covered in Spinulum in Quebec a few years ago. It was probably 3 or 4 acres of almost a monoculture of Spinulum. So they can be quite successful. So the next group, so there were 3 families of lycopods, the Lycopodiaceae. The second one is the Selaginellaceae. We do have a few in the state, not many, but they're distinctive from the Lycopodiaceae in that rather than having helically arranged leaves, they have leaves in either 3 or 4 ranks. Most species have leaves in 3 ranks, not 4. Some have 4 ranks. I'll try to show the 3 ranks in this photo, but I confess this photo on the upper right is actually of a species in Puerto Rico, but I thought it showed it really well. And so I'll try right, over here. There's a leaf running down the left flank. There's a rank of leaves running down the right flank. And then there's a row of leaves right down the center. And the leaves running right down the center in this particular species come to a long awn-like tip. You can kind of see the awns sticking out here or there. But those are the 3 ranks. There's usually 2 lateral ranks and then a dorsal rank. Or sometimes instead it's a ventral rank, but there's usually 3 leaf ranks. There are some species, however, that have something more typical, such as a 4 ranks of leaves. If you have a microscope and you're super motivated, one trait that distinguishes this family is this tiny microscopic structure on the leaf. It's usually at the base of the leaf, called a ligule, which translates to tongue. Under a microscope, here's the ligule. The leaf was attached sort of over here, and then the leaf goes off, kind of off to the right here. This is just a close-up of this little microscopic tongue, the ligule. There's some fairly good evidence that what that ligule is doing is secreting oils and other compounds that do two functions, really. They retard both desiccation, especially during leaf expansion. They also seem to produce compounds that are have some kind of an anti-herbivory function, although that hasn't been well established yet. Looks that way. Selaginellaceae also does something very unique and very bizarre, and that is on the underside of the stems. So, here's a shoot system with lots of little leaves on it, little lycophils. Is this structure that comes out that you might think is a root, but it's not. It's a rhizophore. It does not have a root cap. It does not have the anatomy of a root. In fact, how it originates out of the stem is not from a pericycle like in a root. It is a very unique structure. However, what it does do is it grows out, it'll eventually make contact with the soil, and then roots, real roots with root caps, emerge from the rhizophore. And so that's what you're seeing here, these two little forked things, those really are roots. But the thing those two roots are attached to is not a root, it's a rhizophore. It's this bizarre intermediate structure between a stem and a root that this group evolved, again, roughly 380 million years ago. The last kind of distinctive character for this group is that they're heterosporous. So some sporangia, they make cones, by the way, I should show it up here. Here's a cone. You can see it's distinct from the rest of the shoot system. In this cone, you'll find some leaves will have a sporangium that's small, and they have small spores inside, hence microsporangium. The microspores inside will develop into male gametophytes. On the same strobilus, the same cone, will be a sporangium attached to a different leaf that has large spores, usually only a few, typically 4, sometimes 8. This megasporangium produces megaspores, and the megaspores develop into female gametophytes. Obviously, a plant like this, it's going to take a spore of each type landing pretty close to one another in order to successfully fertilize and produce another plant. So how about a couple of examples from Michigan? On the left is Selaginella eclipse, which ironically, I have not personally found in Michigan. I've tried, but I couldn't find it despite its frequency. You can see it has a rather odd, somewhat disjunct distribution. It's fairly common in the southern tier of counties, and then it kind of reappears sort of along the border of the tip of the glove and into the UP. Kind of peculiar distribution, but it is very much associated with wetter habitats with a wetness index of -3. So it could be that just in the habitats it's occurring in, there's so much other vegetation, I'm just missing it when I'm looking for it. Sort of at the opposite end is Selaginella rupestris, which definitely occurs in drier, tends to be sandy or very rocky sites. And as you can see, has a wetness index of 5. So it's at the other end, highly tolerant of very dry conditions. I like these two species as examples for you because the species on the left, Eclipse, has the 3 ranks of leaves, one on the left, one on the right, and then one dorsal rank. Selaginella rupestris is a great example of a species that has 4 ranks. And so it tends to, the shoot system takes on this more terete or squarish appearance as a result. These are among the more, these 2 species are among the most common of the Selaginellas in Michigan. So they're also good examples for that reason. So the third and last family within the lycopods are the Isoëtaceae. And the Isoëtaceae, make this bigger for you. The Isoëtaceae are more or less strictly aquatic or semi-aquatic plants. And they are the last remnants of a kind of a mighty lineage. The first forests of the world, the Carboniferous forests, the forests that dominated throughout the Devonian and the Carboniferous, forming, the vast majority of our coal beds throughout the world, were actually comprised primarily of members of this group. So the forests were forests of Isoëtaceae. And so they have this kind of great history, but all that's left of them now are these, funny little characters you find in wet spots. But they once ruled the world and reached, easily, that's actually an underestimate, but easily 30 meters tall. We have them all. If you ever stop by Grand Valley, I have a large fossil collection. I'd be happy to show you a ton of these guys. But they tend to have I'll start with the bottom photo. They have a woody little body. So here's the little corm. And by woody, I do mean woody. They're capable of secondary growth. They put on it's mostly secondary cortex. So they are making secondary xylem, but in this loosely arranged, rather feeble effort. So there's some secondary xylem in here, but most of the secondary growth is actually just cortex. It's a whole lot of densely packed, rather highly reinforced parenchyma. And it results in this kind of knobby little thing. Usually those are pretty small, maybe the size of your pinky nail, or I don't know, maybe your middle fingernail is a big one for Michigan anyway. Coming out the top are the lycophils, or the leaves. They can easily be mistaken, especially for a rush, because in cross-section, the leaves have a squarish or rounded appearance. So people will often confuse them with a sedge or a rush, but in fact, not. And I think probably the thing to go looking for is that corm, that woody little corm down in the water. The odd thing about these guys is that there are what appear to be roots sticking out the other end and kind of penetrating into loose substrate. These plants are not able to extend their rooting system into anything very firm. So they have to be in loose gravel, soft silt, something like that, to gain purchase of the substrate. The funny thing is, those aren't roots, they're leaves, believe it or not. We know this from the fossil record. The ancestor of the Isoëtaceae, early on in the evolution of this lineage, they did have roots. They lost them. In fact, those giant 30-meter tall trees I was talking about, they did not have roots. They had the same game that this rascal does. What was penetrating down into the substrate was, believe it or not, a shoot system with leaves coming off. And it was just geotropic, sort of oriented gravitationally downward. And the leaves on those shoot systems acted sort of like root hairs and were the absorptive surface for the shoot system. The fancy word for those, if you're curious, is called a rhizomorph. The rhizomorphs of these ancient lycopods are shoot systems. Again, if you ever stop by, I can show you lots of fossils that document that evolutionary process. It's totally bizarre, but the point is, what's sticking out at the bottom of Isoetes, at the bottom of the corm, the brown things you see here, are actually leaves, not roots. They don't have a root cap. They're not roots. The leaves on Isoetes, I should mention, can be rather long. I've seen tropical species that had, 40 centimeter leaves. Here in Michigan, you can count on them reaching, at least, the bigger ones, maybe 10 centimeters. It can be good sized leaves. Like the Isoëtaceae, they have a ligule, that little tongue that's at the base of the leaf. Which tells you, let's see if I'm going to go up a couple of slides, bear with me here. It tells you why the Selaginellaceae and the Isoëtaceae are linked together as a clade that is distinct from the Lycopodiaceae. It's because of this, the presence of a ligule. It's also because both families, are heterosporous. That is, they make big spores that become female gametophytes, small spores that become male gametophytes. Although I think of them as strictly aquatic, there are some species that will occur in in fens and other habitats that are inundated for at least a good period of time. Those tend to be species that are somewhat ephemeral. That is, the leaves are not up throughout the entire growing season, and so they're easily missed. They tend to kind of come out in the spring and make it through maybe midsummer, and then they disappear. Strange group of plants. Now I'm moving over to what we at least these days call a fern. And they all belong to this mouthful here, the Manilophytes. It's the worst name in the world. I'm not going to even torture you with how it got the name. It's just an awful name. A better name is the fern clade. And it includes, again, these 5 families, 2 of which that don't occur in Michigan. So I'm going to focus on the 3 that do. So we're moving over. These plants, of course, also have roots. They also have leaves, but they've evolved leaves and roots completely independently of the lycopods. And first up is the Ophioglossaceae. There's a couple things you can look. There's really, crudely speaking, 2 genera to think about in Michigan. The Botrychium group, and by that I mean, as you can see, the 3 genera at the top, they're Botrychium, Botrypus, and Septridium. The reason I call them the Botrychium group is that when I was a graduate student learning my ferns, they were all Botrychium, and then more recently through molecular work have been broken up into separate genera. But they tend to have a similar look. And the similar look is that they have a short, erect stem, very short, erect stem, like, I don't know, 2 centimeters. Very thick roots. In fact, here's an example of a root of Botrypus virginianus. Very thick, I mean, bizarrely thick roots. And notice that you don't see any root hairs. That's not because I was careless in how I cleaned them off. They don't produce root hairs. They've evolutionarily lost the ability to produce root hairs, which you might know how they're making up for it. They are obligate mycorrhizal symbionts. They absolutely have to, early in their establishment as a small plant, form a mycorrhizal association. And if that mycorrhizal association fails to form, the little sporling dies. And of course, if there's anything that disrupts the mycorrhizal community, it will tend to spell doom for the local population of Botrychium. So you can't find root hairs there because they've lost them. Another funny thing about this clade is that they produce, of the Botrychium Botrypus Septridium clade, I mean, they produce only 1 compound leaf a year. 1 leaf, that's it. If that leaf gets eaten or smashed or whatever, that's it. They may produce a leaf in the following year if they have the reservoir to do it, or they just die. 1 leaf. And they present their sporangia on a stalk. So here's the 1 compound leaf, and coming out of the base of this leaf, because the stem is underground, is this large presentation. Sometimes these are referred to as the grape ferns, or sometimes the rattlesnake ferns, usually grape ferns, because this particular individual has already released its spores. Had it still, its spores within the sporangia, they would have looked like a cluster of grapes. And so they'll often get the name of grape fern. The thing to know about this little stalk is that really it's two leaflets. It's the basal two leaflets of this one compound frond have fused together evolutionarily into this stalk. We know this because there are some members of the group that still have them as two separate leaflets, plus the fossil record shows it very nicely as well. So what looks like maybe the continuation of a stem isn't, it's just the basalmost two leaflets. Again, something to look for. And obviously, despite this dependency on mycorrhizae, a very successful group, Botrypis is found in virtually every county in Michigan. It's hard. Let's see, is that Ionia County? I have a hard time believing that it's not there. It looks like from this map, that's the only county not reporting it.
It's there.
Oh, I hear somebody. Well, please do interject if you have insights or something to add. I'm still in the Ophioglossaceae. The other sort of subgroup within this family is the genus Ophioglossum. So these two, this clade and this, they're in the same family. They're very closely related. And the big difference is pretty obvious. Ophioglossum makes a simple leaf instead of a compound leaf. But just like the prior clade, Ophioglossom also produces a pair of fused leaflets that will present a sporangium at the tip. And I ran out of time to find a good picture in my library of pictures to show that, but I've at least got this graphic. Unlike Botrychium, where the stem is always erect with fat roots, Ophioglossum also makes these fat mycorrhizal dependent roots, but some species have erect rhizomes, stems. Others have these wandering, laterally growing stems. So it can occur either way. The most common Ophioglossum in the state is Ophioglossaceae pusillum. Not easily found. Took me forever to find one, but occurs in a number of counties in the state, at least from my experience, just not in very great abundance. You have to really be wanting to find one. The wetness index is a -3, so clearly it is highly dependent on moisture. Ophioglossaceae pusillum, believe it or not, it often will occur as something as an ephemeral, where along beaches. So I've even seen it along beaches where it comes up and is around for a few months until it gets hot and dry. So I suppose by mid-June, it's gone again. So it's one of these species where the time of year is kind of critical for finding it in some habitats. In other habitats, it's up throughout the entire growing season. Something I wanted to point out is here, Ophioglossaceae is clearly demanding of rather wet conditions. The prior clade, they tend to be much more upland oriented, right? With the wetness index of 3. So here's the drier kind of clade within the group. And this is the, quote, wetter clade within the group. So we're still in that, fern clade, and I'm leaving the Ophioglossaceae and moving to the next family, the Equisetaceae, the horsetails, right, and scouring rushes. I'm sure all of you know them well. So I'll kind of breeze through them because I'm guessing you're all very familiar with them. One of the distinctive traits for the group is that the stems tend to be hollow with a very large pith canal, which in this microscope slide is just at the edge, the bottom edge of the photo. many species will have two more canals, a canal within the xylem, which is known as the molecular canal for whatever it's worth. And there's often a series of canals within the cortex known as the carinal canals. But the point is, of course, the group has a lot of canals. The canals are pretty obvious. They facilitate gas exchange, particularly in habitats where the underground rhizome system is perpetually submerged, because the rhizome system also has those canals. So a way of getting air down there. It does have leaves, and the leaves are, in this clade, minute. They're whorled in their attachment, and they're fused together into a collar. So here is a collar of those leaves, and right here is the tip of one leaf. And so this is one leaf about right here. This is a separate leaf about right here. And you can see, almost make out a kind of a dark green seam about right there. They're fused for what, maybe 80% of their length. And then the tips become free of one another. If you go back far enough in the fossil record, those leaves are completely unfused and separate. And so it's a derived condition, this fusion into a collar. That means the things that are coming off on a horsetail, those are branches, not leaves. They're stems. Those stems also have whorls of leaves on them. And here's a whorl of those little tiny leaves right there. You can even see the tip of one leaf here. I can just make out the tip of the next leaf right there. So those things coming off on the sides are branches, not leaves. This group makes cones. In fact, there are no exceptions. They all make cones. I should mention that back in the Carboniferous, during those coal forming forests, there was an apple tree-sized tree that was a member of this group known as Calamites. Probably even a little bit bigger than an apple tree. So this clade has a kind of a great history as well in terms of once forming trees and being important components of a forest. No longer the case. If you get really geeked on this group, something you might check out is the spores have these little elaters. These little they're parts of the spore wall, protein structures that will split and dry out and form 4 paddles. And if you breathe on a pile of their spores, they kind of do this dance for you. The elaters are hygroscopic, and they absorb water and then lose the water, and they thrash around. It's a mechanism by which the spores can kind of work themselves out of the cone and then catch the air and fly off. Teaching a college class, students tend to be amused by it. So here's this picture on the right is actually of a species from Michigan, Equisetum telmatia. Pretty big, one of the biggest equisetums on the planet. Can get easily taller than over 6 foot for sure. They're capable of getting quite large. But in Michigan, it's quite rare. To my knowledge, it has only been reported in the Keweenaw. So you'll have to go up there if you want to find one, or out to Humboldt County, California, where they definitely get 9 foot tall, because I've walked through them. Clearly a water demanding plant with a wetness of -5. Another, I think, great potential indicator is Equisetum fluviatile, and it also has a wetness index of -5. This is not my photo. I don't have one of the species, but it is almost purely restricted to lake shores of one form or another. I've never encountered it away from a lake. It's easy to identify because it's notorious for what a wimp it is. It falls over really easily. The pith canal is so large that it takes virtually no effort at all to cause the stem to collapse. And you can kind of see the evidence for it in the photo here, where the Equisetum closest to the water, any wave action at all, and they'll just collapse. The ones in the back are standing up due to sort of group effort, kind of mutual groups support of one another, because no one stem could possibly hold itself up on its own. It's a notoriously sort of flimsy Equisetum. This one's, I hope these pictures work. This is Equisetum sylvaticum, also capable of growing fairly tall. In Quebec, anyway, I've seen it reach, oh, I don't know, chest height, maybe 4 foot. Pretty capable of getting good sized. It's easy to identify. In this photo on the right here, you can see the stem kind of growing up towards us. It looks brown. It was broken. And coming off the side is a branch, right? And it's going to have those whorled collars of leaves. I'm pretty sure that's a whorl right there, that little white spot. The point is that this branch branches. Here's a branch coming off of this branch. It's the only Equisetum that does that. That is, it produces secondary branches. I've tried to blow up that photo here. So again, here's the brown stem that's been broken off. Here's the primary branch coming off. Here's a secondary branch coming off of that. So it's capable of secondary or even tertiary branching. So easy to identify. Sylvaticum is the only one that does it. And like Telmatia and Fluviatile, it's definitely a species that requires persistent moisture. So with a wetness index of -3. How's everybody doing? Can I answer anything, or does anyone want to toss in some personal observations or take a potty break? Well, I fear I'm throwing too much at you, so please do let me know if you want me to kind of skinny this up. Okay, I'll soldier on. Here's, of course, the most common of the Equisetums, Equisetum arvense, and it's literally the common horsetail. And I think its most distinctive trait, in my mind, is the reproductive shoot system, which is ephemeral. It only comes up in the spring. It is brown. It does not photosynthesize. So it's achlorophyllous, right? No chlorophyll. And that doesn't get very tall. These come up slightly before the vegetative stems come up, which I have on the far left. These tightly packed vegetative stems eventually expand out into the more diffusively branched plant that you and I know and love. But for me, arvense, of course, I know I'm around arvense without much effort when I find these fertile shoots without any chlorophyll. Some members of the Equisetaceae don't branch, and of course we call those, the nickname is typically scouring rush. Although I grew up in Colorado, I called them scouring rush. My wife, who is born and raised in Grand Rapids, calls them snake grass, which I'd never heard that before, but apparently another nickname is snake grass. This is the most common of the scouring rushes, Equisetum hymale, and it actually does branch sometimes. I took these pictures down here in the Byron Center area, but it clearly has a strobolus. Once this strobolus dehisses all of its spores, and they'll usually turn brown and literally fall off. In fact, I'm pretty sure right here is where as a stem where the strobolus has fallen off. Sometimes right at the apex here, they'll make little branches. They're wimpy little branches. They never become very stout. you can sometimes find these sort of half-hearted attempts at branching near the tips of the stems where the strobilus has fallen off. Because of course the strobilus consumed the apical meristem, and so there is going to be no further growth. You may know this, but Equisetaceae has a funny trick up its sleeve. At each node, so here's a node, of course, and here's a collar of those. I'm sorry? I heard a question. At this node here, there's this fusion of leaves. Right here at this node, there's also a intercalary meristem, meaning that even if the apical meristem dies, this internode here, from one node to the next, can lengthen because of the activity of the intercalary meristem that sits just above the node below. So these internodes will get longer and longer over time. Even if the tip has died, it turns out the stem can still get longer because it has that hidden meristem at each and every node. So one thing you can notice is as you travel down an equisetum, any equisetum, the nodes get progressively longer and longer. And this node will eventually get as long as the basal node. It just is going to take it time for that intercalary meristem to produce it. Last but not least, of course, are quote, the quote, true ferns, the Polypodiaceae. A lot of these photos here are not of species in Michigan, but they are the second most successful clade of plant life on the planet. They are far more successful than the gymnosperms. They're second only to the angiosperms, the flowering plants, in two respects: number of species, but also in terms of worldwide distribution. And I should say, and morphological diversity. So most gymnosperms, which are what, the ginkgos, the cycads, and the conifers, are not very diverse morphologically. Within the gymnosperms, there's only one vine. There are no annuals. All gymnosperms are woody. That's not the case for the true ferns, the Polypodiaceae. There are minute, ephemeral, floating plants such as Azala right here in this middle picture. There are tree ferns, although they're not making secondary wood. They're not making a secondary xylem. They're not making wood, but nevertheless, they do form tree structures. All sorts of strange herbs like this. Believe it or not, this is a blechnum in Puerto Rico. They can form vines, much like brambles. This is Odontosauria, also from Puerto Rico. But notice it has prickles like you'd expect on a rubus, on some member of the Rosaceae, right? Actually has prickles. They'll just tear you to shreds. They're horrible. You can tell it's a fern, all right? It's unfurling, compound leaves, the whole bit. The point is that the morphological diversity, the anatomical diversity of the Polypodiaceae is quite substantial and second only to the Magnoliales, the flowering plants. Flowering plants, there's about 300,000 species that have been documented. For the Polypodiaceae, there's about 15,000 that have been documented. There's only about 700 species of gymnosperms. Not very impressive, right? In comparison. Well, why are they, just a brief tangent, why are the polypods so successful? Well, it turns out the best thing that ever happened to them was flowering plants. As flowering plants took over the forests, prior to that, the forests were composed of conifers, cycads, ginkgos, and conifers, but mostly conifers during the late Jurassic, early Cretaceous. The angiosperms steadily marched in their competitive takeover of the world's forests. And it turns out that as the angiosperms were doing that, the polypod ferns were pretty much doing the same darn thing. Long story short is that the polypods evolved unique photosynthetic mechanisms to take advantage of the red wavelengths that are left behind after light has penetrated an angiosperm canopy. The wavelengths that reach a forest floor are rather low energy red wavelengths. And it turns out polypod ferns have evolved a really interesting phytochrome system that allows them to be very good at harvesting red wavelengths of light. And so they have taken over the understories of flowering plant forests. They also did something else, and that is they climbed up the trees, the flowering plant trees. So, for instance, in Puerto Rico, but this is true on anywhere you want to go in the tropics, the majority of plants growing on epiphytes growing on the trunks of trees are ferns. So, of course, there are bromeliads, the pineapple family. There are orchids and so forth. There's all sorts of members of the coffee family, the Rubiaceae, et cetera, growing as epiphytes. But the community of epiphytes is dominated by polypod ferns. And so they've not only successfully figured out how to compete and thrive in that low photosynthetic energy of the forest floor, but how to kind of run up the trunks of the flowering plant trees. And that's why they are so successful today. So I wanted to maybe mention some useful anatomical traits for identifying species and some useful reproductive traits. The useful anatomical traits include wiry roots. So remember, the Ophioglossaceae had those really substantially fat, thick roots. All of the Polypodiaceae, without exception, have really tiny, wiry roots. Even those tree ferns that I showed you, their roots, in fact, this is the root mantle of a tree fern. They're always really tiny, itty bitty, and very brittle, heavily lignified root systems. One thing that you can key off of is how diverse the leaf and stem morphology can be. But before I go into that, I should mention that without exception, the leaves or fronds of the Polypodiaceae unfurl as a fiddlehead. There's a few angiosperm families that do that, but the Polypodiaceae exclusively do it. There are no exceptions. The three little diagrams on the right are merely meant to visually illustrate that you can get kind of what you'd expect, the kind of Joe Blow erect fern. It doesn't creep around on the forest floor like Christmas fern, if you're familiar with it. Or something that wanders around where the stem is underground and it's sending up leaves. Of course, these can be real mischief makers like Pteridium aquilinum, right? Bracken. That's what bracken is doing with a rhizome that can be, 18 inches below ground, which I'm sure all of you know makes bracken very difficult to control. You can burn the grassland that bracken has invaded. It'll just come right back up because the rhizome is completely protected. But this group have all sorts of bizarre stem leaf relationships. So there are examples, including here in Michigan, where the leaf is indeterminate. It doesn't cease growing, right? We tend to think of leaves as determinate structures. They have an end point, they stop growing. And there's a leaf. Well, that's not necessarily true for polypod ferns. Some of them make leaves that do not ever stop growing. I'll show you an example, admittedly from Puerto Rico, but this is Dichronopterus flexuosa, and this branching light green thing you see here, that is not a stem, that is all leaf. The stem for this plant is underground. Everything above ground is leaf. the leaf has unending growth. It just keeps unfurling and unfurling and unfurling, and it never stops. And of course, then leaves emerge, but it just doesn't stop expanding. And so it's a vining kind of structure that sort of defies our sense of what a stem and a leaf is. But they can take over an entire hillside. This is a large landslide on the road up to El Yunque Peak in El Yunque National Forest in Puerto Rico. And all of that vegetation there is Dichronopterous. It can not only take over disturbed locales, it can completely arrest succession so that seedlings of trees cannot penetrate what could be a 6 or 7 or 8 foot thick mat of these leaves that are just growing over each other and over each other. And it just utterly arrests succession. We have an invasive here in Michigan in the genus Lygodium. It's a big problem down south, especially in areas like Georgia and South Carolina, but it has been recorded over in the northeastern corner of the state. And it too, Lygodium, has an unending indeterminate leaf. So it'd be one way to recognize that invasive is this kind of indeterminate leaf. The leaf can even branch. So depending on what key you use, you get tortured when you key out the polypods because of all of the lingo that was developed back in the 1800s. And you know how clicks are. Rather than using words like leaflet, to this day, pteridologists will use the word pinna. So there's this sort of archaic language that, due to the quirky culture of subgroups of science, have kind of retained these cumbersome words, but nevertheless, I've labeled them for us here. This is supposed to be a twice compound leaf. And so we have a leaflet that is composed itself of many leaflets. So this whole leaflet here is usually referred to as a pinna. Pinnae is plural. Each leaflet, if there is one, of a leaflet, right? A secondary leaflet is not referred to as a pinnule. Rather than calling the stalk of the leaf a petiole, they're often referred to as a stipe. Don't shoot the messenger, please. And there, I call this the midvein or the internode of the leaf. Not sure what corresponding language to use, but this is often known as the rachis. And these words are relevant in that often the key is guiding you to something funny about the rachis. For example, in Puerto Rico, there are species where the rachis is winged. It actually has little flanges sticking out sideways out of the stem. So the words can be useful in directing your attention, but they're rather tedious. And of course, the midvein or stalk, well, not stalk, the stem, I guess, of a leaflet is known as the rachilla. In case you ever come across a key like that, I thought this might be useful to you. The picture on the right is simply meant to say things can get pretty crazy with ferns, where here we have a leaflet, this is a pinna, with pinnules, but then the pinnules can themselves be composed of tertiary pinnae, right? So polypod ferns can get quite lacy in appearance because of all of this multiple levels of compound structure of the leaf. And it can get even worse. For instance, you can get a complex compound frond where the basal leaflets are compound and themselves have pinnules that are compound. And yet on the same leaf, the next leaflet up is no longer twice compound. So, and then you can go up even higher still, where now a pinnule, a leaflet, is entire. In fact, pteridium, bracken fern kind of does that. It's playing sort of that little game right there. And you can then get everything down to just good old fashioned pinnate leaf to pinnatophid, meaning the leaflets aren't quite distinct, to simple leaves. Just as a point of curiosity for you, the fossil record shows that the earliest members of this group had pinnate leaves, and then they've either elaborated and may become more complicated in their leaf structure, or they've become increasingly simplified in their leaf structure. But this is actually the beginning point for the leaf evolution of the clade. So useful reproductive traits. I feel like I'm missing a slide here. Let me double check. No, I am not. Okay. Useful reproductive traits is that the polypod ferns have a very distinctive sporangium that reminds me of a lifesaver lollipop. If you remember the lollipops, and they have a ridge on them. If you're my age, you know what I'm talking about, the Tootsie Roll Center. That's what these remind me of anyway. There is a stalk on them, just like on the lollipop, and then there's this round structure at the top, which is the leptosporangium. And the leptosporangium dehisces, releases its spores by the action of this ring, this mohawk structure called the annulus. The shape and orientation of the annulus is diagnostic for various species, but fortunately, for all Michigan species, you really don't need to pay attention to it. Long story short, the annulus, when it loses water, it will kind of expand back and rip the bag open, rip the sporangium open, and sort of like a bow and arrow, the tension is created in that annulus as it's pulled back through water tension. Finally, the tension in the walls of the annulus exceeds the strength of the bonds in the water molecules, and it snaps forward and ejects the spores out into the air. So the annulus is just the mechanism by which spores are thrown out into the air. A cluster of these leptosporangia is known as a sorus. I'm guessing you probably know that. And often a sorus, a cluster of sporangia, is protected by this structure, the indusium. This particular indusium is shaped like an umbrella with a stalk coming up the center and then fanning out. This indusium was once much larger. It has shriveled up to get out of the way of the sporangia, because the sporangia are now ready to dehiss. And so it kind of contracts up. So this leptosporangium, this lollipop structure, is diagnostic for the polypod ferns. The other groups, the Equisetaceae, the Ophioglossaceae, the Lycopods, don't have these lollipop structures. If you have a good hand lens, if you look at a sorus, you can definitely see these. They're not that hard to see. So some more useful reproductive traits. Again, a sorus is a single one of these clusters of sporangia. Sori would be plural, so here are sori, two of them. So there's lots of sporangia underneath this. This is a sorus. There's hundreds of sporangia here. They're black, this species, when they're ready to pop. This is the kind of reniform indusium, kidney-shaped. You can pretty much figure out where the stalk is on the thing, and it will shrivel up and get out of the way to let those sporangia dehiss. So when it comes to sori, are very useful for identifying species. First of all, some species make them, and some don't. So this is a genus that doesn't make an indusium. It has sori for sure, right? One sorus, two sorus, three. but the sori don't have an indusium. There's no protective covering during the development of the sporangia. The $10 name for that is to be X-indusiate. The thing to really key off of in my mind is the shape of a sorus. So here the sorus is round with a reniform shaped indusium. Here the sorus is crescent shaped. I'm trying to think of it. It's the best I can come up with. It kind of reminds me of a crescent. Here that's linear to somewhat crescent. They can have different shapes, linear, crescent, rounded. And those are very much diagnostic for different genera. Also, where the opening is. So for instance, in this Dryopterid here, clearly the sporangia sort of face out towards the margin of this pinule, because this is a leaflet of a leaflet. Okay. Whereas check this out over here in this is an asplenium. The sorus is not only linear, but you can see that the opening is towards the midvein. They open towards the midvein rather than towards the margin. So by paying attention to three things: shape of the sorus, does it have an indusium or not, and what is the orientation of the indusium, if it's there, really will get you really fast to a genus, or even, not to a species, but to a genus for sure. And we'll look at some examples of this in a minute here. Maybe another thing to notice is, for instance, this is Dryopteris marginalis, if any of you know it. Notice that you can tell how it got its name. The sori are right on the margin of the leaflet. Here, the sori are they're far from the margin. And so just looking at those little things, shape, indusium, orientation of opening, where are they relative to the edge or the center of the leaflet, really will tell you a lot really quickly. The same thing for the protective structure, that umbrella, the indusium. Is it peltate, like we saw here, where it's like an umbrella and the stalk is in the middle? Is it reniform, where the stalk is still in the middle, but you can tell there's a cleft? Or whether the flap is linear or not, will really help you identify various polypods. So maybe some examples are coming up. But last but not least is a couple of quick vegetative traits. Throughout much of the world, I would need to give a talk where I carry on about vein patterns. The venation patterns of the leaves are extremely important for identification. So too are these structures called scales, which I'm not going to go into because we don't need them for Michigan species. But if you're anywhere else, if you're in the tropics, you would absolutely have to be paying attention to those. More importantly, locally, is just some overall structural aspects to the frond. Is it simple? Is it pinnate? Is it have some kind of complex compound structure? Next would be, are the fronds monomorphic or dimorphic? So if I start over here with this lady fern growing on the side by the side of my house, the lady fern here, every frond is potentially fertile. Or another way to say it is, the fertile fronds look just like the vegetative, non-reproductive fronds. There's no anatomical distinction. Or you could have something like the far right here, clearly cinnamon fern, where these spears here, these are just fronds, they're just leaves. Under a scope, it becomes really obvious. But the leaflets, and there are leaflets there, but they're very tiny and curled up, and they're loaded with sporangia, those leptosporangia. And so clearly it's dimorphic. The fertile leaves are visually quite distinct from the vegetative leaves, clearly dimorphic. Or you can get in between things like the Christmas fern. And the Christmas fern, and I'm going to show you another picture of the Christmas fern in a minute, but on a fertile leaf, the vegetative leaflets are quite obvious. They're rather large and flattened out for photosynthesis, but you can tell there's an apical triangle here and a section where suddenly all the leaflets are quite a bit smaller. Seems like a lot of bad design for photosynthesis. Well, those are the leaflets that are fertile. The leaflets below are vegetative. They don't have any sporangia. So this would also count as a dimorphic frond, right? Because a vegetative frond of Christmas fern doesn't have this sudden constriction in the size of the leaflets. I'm going to show you another photo of that for Christmas fern. It's coming up. So I thought I would go through a few important species, and we'll do it from on a wetness gradient, starting with sort of being out in the middle of a lake, and we'll work our way to an upland. We do have a zola in the state, also known as mosquito fern. As you can see, it's been reported in 2 counties. So my gut feeling is with climate change, we're going to see this really take off like duckweed. I think Michiganders are going to view this plant the way we view duckweed, right? As a real annoyance. It's going to change water quality, and the reason is that it forms a symbiotic relationship with nitrogen fixing Anabaena. So and some of you may know this, it's often known as green manure, because in Southeastern Asia and in California, farmers will deliberately sow this onto their rice paddy fields. And so rather than using manure or fertilizer to fertilize their rice paddies, this plant will increase the nitrogen availability, which is often limiting, right, in plant growth, within the rice paddies. So I think if this really gains purchase in wetlands here in Michigan, we could see eutrophication issues pop up. Now, I realize that I've got to then think about phosphorus, but I think a double hammer of this on top of other forms of eutrophication could become a new problem for Michigan. Anyway, it's a floater. And by the way, I'll try to, this right here, that's one plant. So you're looking at, I don't know, a few hundred plants, or at least a few dozen. If you were to scoop them up and put them on your finger, you'd have at least a dozen on your finger. These are tiny little plants with little root systems, the whole bit, dangling down. And also ancient group, and responsible for a major shift in climate some 50 million years ago. There was a zola catastrophe up in the Arctic Ocean. You might look it up sometime. A zola catastrophe, Arctic Ocean. You'd be surprised what an effect this plant had on global climate. Moving kind of, heading towards shore, one of the more notable species and more common, right, you can tell by the map, is regal fern, Osmunda regalis. My bet is most of you know the plant. Tends to form in shallow water, bayou-like conditions, and the root system is erect, or excuse me, the shoot system is erect, and eventually builds something of a mound. So they will eventually sort of create these emerging micro islands within a submerged habitat. They definitely alter sort of they can potentially alter the hydrology of a wetland. I'm going to show another member of the group. The way to tell them apart is pretty easy from the distance. Notice how kind of smooth and plump the leaflets are on Osmunda regalis. To me, it's very distinctive. I don't really have a descriptor for it, but there's something about that to me that from afar away, I know the regal fern when I see it. Next up, right, we're moving from wetness index of -5 to -3 is Tholipteris palustris, which I'm sure all of you know. It's just everywhere. Impressive plant. It's a phalanx colonizer. So that could all be one plant with a branching stem system that's just ramifying and branching hundreds and hundreds of times down in the muck of that shallow water. This is a boardwalk at Ludington State Park. So I'm sure you know the place well. Tholipteris, the whole genus is really easy to identify. Tends to have twice compound fronds, not always, but usually. But the leaflet margin is always hairy. So the edge of the leaflet, there'll be these little hairs sticking off. And that's diagnostic. Tholipteris is the hairy family. And of course, can form very dense stands. Sort of moving ashore, as it were, we come to Osmundastrum cinnamomea, or cinnamon fern, a close relative of Osmunda regalis, the regal fern, but they can be told apart by a variety of ways. But one is that the leaflets of Osmunda are lacier. They're not as entire. I'll jump back up just for a sec. Look at how the leaflet is just smooth and wide and entire, very distinctive from the leaflets of Osmundastrum. I have to confess, to this day, Osmundastrum, cinnamon fern, and Matusia struthiopteris, ostrich fern, especially in the spring, right after a long winter and my senility kicks in, I can't tell the two buggers apart. They drive me nuts. Their reproductive structures are quite distinct. So let me mention those for a minute. The spears of the cinnamon fern I've already talked about, but on the ostrich fern, this is still unfurling, but it's going to make this flattened out display. It's a frond, it's a fertile frond, but it's going to make a flattened out display that turns a dark purple. In fact, I have one right out my window here. I should have taken a picture of it. This is the same one that I took the picture a couple of weeks ago, and it hadn't matured yet. They turn out to be these flattened purple displays, and hence the name ostrich fern. It almost looks like maybe the flattened display of an ostrich tail. But vegetatively, the two, to me, look a lot alike. And to make my case that it's not entirely senility, is here's a leaflet from the ostrich fern immediately above the leaflet from a cinnamon fern. These two plants are growing right next to each other in my front yard. And for the life of me, they look an awful lot alike. And here's what I key off of to tell them apart. Assuming the reproductive structures aren't available, there's a good hunk of the summer when they're not. But you can't tell who's who. And that is, the cinnamon fern gets its name by these little clusters of cinnamon hairs that are at the base of a leaflet. So at the base of this leaflet, where I tore it off, there was a cluster of cinnamon colored hairs. Two problems, sort of. One is you need a hand lens. I can't see them with my naked eye anyway, but I need readers now. You need a hand lens to see that tuft. And the second is, they're not always that cinnamony, right? They often are kind of a cinnamon color as they mature. But as you can see from my photo, often they're kind of a translucent color and not very easily seen. But there is always a tuft there if you look for it. Oh, I should be careful here. There's usually a tuft. The tuft of hairs can fall off over time, making the whole diagnostic worse. Here's the thing I go for. I'll see this is not in a key. I'm just telling you what I have learned to look for. I'll see if I can convince you that I'm onto something. Here are these two veins at the base of this pinule, right? Because that's a leaflet of a leaflet. And this innermost vein arches out, right? It gets a little bit wider, but then when it comes back in, the distance from here to here is roughly the same distance as from here to here. So despite the arcing, at the end of the day, the distance between veins is held fairly constant. Not Matusia, not the ostrich fern. If you look, the veins tend to be held fairly constant till you get to that innermost vein. It too swaggers out, but when it swings in, it produces a much more narrow distance between the two veins. And here I would argue that I'm being proven again. This is what I key off of to tell them apart. If it's, say, mid-June and the cinnamon colored reproductive structures have collapsed and rotted away, or mid-July maybe, and the ostrich fern is still months away from producing its reproductive structure because it does it in the fall. That's what I go after to tell them apart. Otherwise, in my own front yard, I don't know who's who, mainly because the ostrich fern keeps spreading. So as new plants come up, I'm not sure who they are until I do my little vein trick. That's what I use when I don't have the reproductive structures in hand. And I should also note that Matusia is ranked
Thank you.
as by MNFI
Already after some tree has
as
never
having a wetness index of 0. So apparently it tends to be in somewhat drier habitats.
Love you.
That's not my observation. I thought maybe I heard a question. We're getting into drier habitats now. Two species that can easily be confused with one another is the sensitive fern on the left, Onocleas sensabilis, and on the right, Phagopteris hexagonoptera. Phagopteris is a thalipterid, meaning the leaflets have hairs on them. It's part of the hairy group, whereas Onoclea is in a completely different family, the Ophioglossaceae actually. And, but superficially, I find them hard to tell apart. Again, especially if I'm rusty, it's been winter, I haven't been botanizing for months. When they first come up, I'll confuse them. The more you look, the more you can find some distinctions about them. For one thing, Phagopteris tends to occur in wetter habitats. I associate Onoclea with lake and stream margins. I've never personally encountered it away from really wet areas, whereas I have encountered Phagopteris in just moist forest. The other thing to tell them apart is that Onoclea produces a reproductive structure, which again is just a modified leaf that looks like a cluster of grapes, kind of like the Botrychiums. In fact, it's going to turn purple when it's ready to release. But Onoclea makes lots of fronds. You're not going to confuse this with those Botrychiums that only make one frond per season. The fronds of Onoclea are much larger, and the plant has an underground rhizome, so there's lots of leaves around. They're easy to tell apart. But I think these are two to, if you're trying to learn your ferns, these are two you're going to want to learn to separate apart. One of the things I used to key on, but I find it increasingly unreliable, is that the lowermost leaflets, the pinnules, the leaflet of the leaflet, it tends to be deeply dissected, right? You can see there's a cleft that goes way in, whereas the clefts on the lowermost pinnae aren't that deep. That's always true of Onoclea, but I've come across Phagopteris where the cleft wasn't that deep, but they usually are. So that's what I go after. So I think a species of particular use to your group, I would suspect, is the lady fern because its wetness index is 0. Pretty easy to identify, rather lacy looking because the leaves are twice compound. Also, in silhouette, the fronds are obovate, meaning the widest part is not in the middle. It's, I don't know, 3/4 of the way up from where the leaflets begin. Kind of widens and widens till about what, maybe here, and then it narrows up. So it's obovate in its overall shape. That's just really diagnostic for sure for lady fern. Lacy, obovate, lady fern, you're done. And it's very common, as I imagine all of you know. You could always take it a step further. They have these crescent, not linear, they're definitely crescent shaped sporangia when they're fertile. The most common genus of fern in Michigan is the Dryopterids, the family Dryopteris. And here I'm going to emphasize 3 members. They're the 3 most widely distributed and common members. And that's Dryopteris carthusiana, down here at the bottom, Dryopteris marginalis, and Dryopteris intermedia. And I was just going to give you a couple quick tips to tell them apart. Again, by far, this is the genus that you're going to encounter, especially in drier upland forest habitats. So I'll begin actually with marginalis and get it out of the way since I talked about it earlier. Its sporangia occur at the very margin of the leaflet, hence the name marginalis. The indusium is reniform. That pretty much gets you there. There is no other Dryopterid that does that. Marginalis is scored by MNFI as with a wetness index of 3, which surprised me a little bit because I have found it in rather dry dune forest around Saugatuck dunes and places like that, where it doesn't seem like that should be its score, given the drained dune it was on and how high up on the slope it was. Here's its distribution, pretty common in much of Michigan. Next is Dryopteris carthusiana. I took this picture at Ammon Park here in Grand Rapids, if you're familiar with it. Lacy, right? It's easily confused with Dryopteris intermedia. They are sister species. They're very closely related. Worse still, they hybridize. But here's how you tell them apart. It's actually easy once you know what to look for. What we're gonna do is go to the lowest leaflet, the lowest pinna, okay? And we're gonna look at the basalmost pinnule. Ugh, what do I mean? All right, this is a leaflet up here, right? On intermedia. This is its pinnule, and it's basalmost because it's closest to the rachis, the internode, the stalk of the leaf. Okay, so basalmost pinna, and now we're going to look at the basalmost pinnule of the basalmost pinna. I hope you followed that. It's the bottom of the bottom of the bottom. Maybe that's easier. In intermedia, the length of this pinnule is going to be very close, if not identical, to the length of the adjacent pinnule. You can see this on the right-hand side too. Here's the basalmost pinnule, and the pinnule adjacent to it is actually a bit longer. But in Carthusiana, this pinnule, this basalmost pinnule, would be substantially longer, be out here relative to the adjacent pinnule. This pinnule would be longer, it would stick out farther. And that's what I'm trying to say. Well, here's a typo. So this should say basemost pinnules longer than adjacent. Here, it's the same length. So I just wrote the same thing twice. Oops. Intermedia, same length. Carthusiana, it's longer. The bottom of the bottom of the bottom is longer than Carthusiana. Of the three, there's also sort of an alphabetical trend. Carthusiana is ranked as more moisture demanding, wetness of -3. Going down the alphabet to intermedia, we get a wetness score of about 0, marginalis a wetness of 3. So you can kind of trend it out that way in terms of kind of wetness and memorizing who's what. One more species. This is also taken at Ammon Park in Kent County, is Dryopteris goldiana. It is another really easy to recognize species, and that's because the pinnae, the leaflets, are, maybe I should say the pinnules, the leaflets of the leaflets, are tightly packed and plump. I hope that's a good descriptor, right? They're broad and rounded. There's not much of a space between them. In fact, they'll even shingle a little bit over each other. Something very, to me, distinctive about the plump pinnules of Dryopteris goldiana. I'm trying to make this entertaining. We're nearing the end of this. Again, is Philipterus noveboracensis. So remember, there was Philipterus palustris that is associated with really submerged water. Here, this is more of as you can see from this photo, this is taken at Palmer Park in, I guess it's the city of Wyoming in Grand Rapids. But you can see it tends to be very much associated with wetland boundaries. And so I would think a very useful species to know for delineating wetlands. Sort of like Philipterus palustris, it's a very lacy creature, and look for the hairy leaf margins, right? That will always get you to Philipterus really quickly. Right? Philipterus is sort of the hairy group. Christmas fern, one of my favorites. I've done quite a bit of research on it. As I mentioned earlier, it has a dimorphic frond, where the fertile frond, the leaflets that have sporangia are quite distinct from the leaflets that are vegetative. But check out in this photo here, this vegetative frond, you can see that at its apex, there is no constriction. So this entire frond is vegetative. It's not going to reproduce. And then you can see over here, here's a fertile frond, because as we get towards the apex, suddenly the leaflets get so small we almost can't see them. And it's always very abrupt. It's not a gradual transition. There's always this abrupt transition. It's very distinctive for Plasticum acrosticoides. The genus Plasticum is a tough genus, meaning I tend to think of it as something that can handle profoundly cold temperatures for a fern. Ironically, Christmas fern is not found apparently in the UP, and it kind of peters out as we head north. I have no idea why. I'm intrigued by that. I think of it as a fern of drier upland woods. In Appalachia, it is so successful, it can be dense carpets on mountainsides, extremely common, and probably the most abundant fern of the eastern United States, even more so than bracken fern. If you take the entire eastern, I guess half, Mississippi east, there are more plants of Plasticum than of any other species of fern, for sure. There's just countless billions of them in certain states. Another fern I think would be worth memorizing if useful is Gymnocarpium dryopteris. This is a small and very delicate looking little fern. And it's a guerrilla colonist, meaning its rhizome is sneaking around other plants and then sending up a frond. And so you tend to get the fronds intermixed with other plants, making kind of a pretty little scene, I guess. But I think when I find one, I know I'm in pretty wet habitat. In fact, I came across this while hiking in the Olympic Peninsula, or not the Olympic Peninsula, Glacier National Park this summer, and Olympic in the past. It's a species of really of wet and cool habitats. It's a post-glacial species. On the right, of course, is bracken fern. It's more of a phalanx colonist. Sometimes in describing a fern, a key, they'll mention whether they are phalanx or guerrilla colonists as a diagnostic trait. Phalanx is sort of, like a Roman squadron of soldiers marching as a phalanx, right? Rather than guerrilla warfare where you're sneaking in, they're just marching in as one giant army. I thought it'd be useful to bring up those terms because they sometimes are relevant for king out plants, king out ferns. A lot of what I think I've been saying, I have to take with a grain of salt because there are these various wetness indexes. But notice pteridium here has a wetness index of 3. Osmunda cinnamomea, if I remember right, it's -3. But here is the Nordhaus dunes. And that just wonderful and fast, at least to me, fascinating hummocky habitat. And in the on the hummocks, you'll have pteridium that literally is adjacent to these inundated low spots full of cinnamon fern. So obviously, that means we're going from a -3 to a +3 in a matter of like 6 inches. But it's true that where the cinnamon fern is, you can't find any bracken fern and vice versa. So clearly, there's enough of a topography here that the water dynamics are sufficient to create these boundaries. This is also a good slide to mention something of background use, I hope, to you, which is what limits fern evolution, what limits the success of ferns, the reason they're not as abundant as flowering plants, really isn't their reproduction. It's not the whole flower versus spore thing. That certainly plays a role. But far more importantly is that ferns are incapable of opening and closing their stomates, those little pores that are on a leaf that can control, that is, close up to reduce or stop water loss and open up in order to allow carbon dioxide in for photosynthesis, right? That's the trade-off. You open up for carbon dioxide, you lose water. Angiosperms, flowering plants, have the ability to physiologically control the opening and closing of those stomates. And that is definitely linked to the evolution of very sophisticated vascular anatomy in flowering plants. The ferns have never evolved the ability to physiologically open and close their stomates. They've had it a few 100 million years and they still haven't figured out how to do it. And that has limited the evolution of their vasculature, of the cells making up their xylem. And that's really what's restrained ferns to being what we think of as plants that love the wet and the shade. There are, as you probably know, ferns that grow in the desert, but those are adaptations where they basically go dormant for months at a time when water becomes scarce. So that's, with that background, it starts to make a bit more sense that it doesn't take much to go from a species with a wetland index of -5 to the wetland index, a species with a wetland index of +5. So getting really dry, last couple of, this might be my last species, if I remember right, is the aspleniums. Asplenium platymuron is the more common species. They have these linear sori with linear indusia facing towards the mid vein of the leaflet. I don't know why, but it seems to be very much restricted to the lower tier of counties, plus sort of the western coastal counties. I suspect just because they're sandier, and this species is very drought tolerant. Despite the speech I just made, this species is a tough little bugger for a fern and can grow in very hot, droughty, sandy, well-drained habitat. With one exception, it's got to be shady. If it's a sunny habitat that's well-drained and sandy, forget it, you'll never find it. But if it's good canopy, this species can pop up. So it's probably I think of it as the toughest of the ferns personally. A close relative that you'll find on rocks and sometimes on tree trunks is Asplenium rhizophyllum. What's notable about this is not only does it have a simple leaf, but the leaf is capable of cloning. So it will, almost like the stolon of a strawberry, the tip of the leaf can gain purchase on the substrate. Roots will come out, a new stem will grow, and you get a completely new plant, a clone. And then the connection between the original plant and the new plant will eventually wither away, and they will become separate, distinct plants. So this thing's capable of cloning through its leaves, again, because the leaves of polypod ferns are a bit bizarre. By the way, I interpreted this map a few years ago as South Manitou Island. So when I was hiking there a couple of years ago, I spent a horrendous amount of time looking for it. I couldn't find it. Any of you ever find it on South Manitou, let me know. I was asked to give you an assignment, which I think I might be doing this in jest, but this is my next to last slide. Okay, here's an assignment if you want one. I challenge you to try to find one member of each of these groups I've talked about, Lycopodiaceae. The Selaginellaceae, if you can find it, good luck. They're just not that common. Isoëtaceae, Ophioglossaceae. And then I decided to up the game a little bit and have you identify two of the osmundas, the regal fern, the cinnamon fern. Should be easy for you. And then maybe a couple of the dryopterids. Maybe I should have added in there at least one Thelipterus, right? There was Thelipterus palustris, there was Thelipterus noveboracensis. There was also Thagopterus that looked a lot like Onoclea. Those are all in the Pteridophyte, the hairy fern group. So I think I should have added that in there. That's just a matter of kind of wandering around. Maybe on a Sunday afternoon and seeing what you find, right? Almost like a birder, you could start making a trophy list of what you have found and keyed out. That's how I was thinking of that assignment, is trying to get you to think of ferns almost like a birder thinks about birds. Probably more useful to this group is something a little more obvious, which is set up a transect from water edge or even well into the water, in case you can find some floaters, all the way up to an upland location. I arbitrarily decided to make it 10 meters wide. I don't know why. I figured you can easily look 5 meters to the left and right, and you just walk your transect and just key out every single species you encounter. It probably will also help you get a sense of what species you might find useful to commit to memory in terms of wetland delineation or something like that. For your own sake, but maybe for the community's sake, I would encourage you to photograph the entire plant, the entire frond, and to photograph the key reproductive traits. for one thing, we could build a website where you can go to the website and we could look at all the photos everyone's posting and start to sort of have a photo glossary that could be useful. And also, you might as well record the location of where you're encountering each species, regardless of which assignment you do. Because I suspect that a lot of the records for these ferns are within the Michigan flora are out of date. And it would be useful to know, for instance, has Azala spread to other counties? I bet it's going to in the coming years. So my last slide is, I really didn't know what the group needed or wanted to learn. So I just had to kind of go for it. If this, if there's some information you'd rather have heard or today, let me know and I'll try to recraft something for you that I could send to you. But I'm just hoping to get some feedback about whether this was useful to you or not. You're not going to hurt my feelings if it wasn't. I was working off of guesswork. With that, I'll stop because I'm out of time.
Thank you so much. This was really great information for us. And I learned a lot, and I'm excited to go back out to Amon Park or wherever and check out the ferns and look at them more closely and strengthen my diagnostic skills. I should tell you, Gary, that I think usually after these classes, Michigan Wetland Association sends out a questionnaire to all the participants for feedback. And so we'll get those comments back to you. I kind of expect one feedback might be that it'd be really great to have an in-person class at some point. So the education committee can maybe add that to their list of potential in-person classes for the future. So we usually they do that planning late in the year for the coming year. So yeah, so I think we'll get some feedback to you. There were comments.
I was just gonna say I'd be happy to do that. And I know that Bob Hollister presented to the Wetland Association, and he and I have been talking about how we can combine what we've been doing into some kind of a resource, a central warehouse for you guys.
Yeah, we appreciate it. You know, we this is our major way that our members have educational opportunities is through these classes put on by the Michigan Wetland Association. So we greatly appreciate it. You know, we all spend a lot of time in the field, but we don't always have a lot of energy to go a little deeper. And so it's a really great, concise way to get that information. There were a couple of comments in the chat. I didn't know if you were able to catch these while you were talking, but
I missed him.
Brandon noted that he was in Humboldt, California earlier this week, admiring the ginormous horsetails while on the trails. And he thought they were related to ours, but had never seen them like that. He said he's 6'7" and was looking up at the horsetail. So they were huge.
I'm going to guess where he was is actually in the Humboldt woods behind Humboldt State University, which is my alma mater. And they're absolutely colossal back in those woods.
That would be really cool to see. And one other comment, Dan Calla said that North Manitou might make more sense for that one fern that you were looking for.
I've never been to North Manitou. I need to go there.
So my phone's making noises. Does anyone else have anything you want to say before we sign off? Giving you all a last chance.
Thanks for your time. It was a great presentation.
Yes, thank you very much, Gary.
Thank you. I appreciate your time. That was a 2-hour slog.
Oh, it's well worth it.
Well, thank you for the invitation. I'm sure you're all eager to go and enjoy your weekend. So I thank you for the invitation. I hope it was useful to you. And Bob and I are going to keep working on trying to produce some kind of a resource for.
Yeah, and he introduced us to the online I don't know what you call it, kind of database on your Grand Valley. So yeah, if you other folks here didn't do that other class with Bob, if you go to the Grand Valley State University's website and go to the biology department, there's a link to an online resource that Gary and Bob have been developing, which is just a quick guide to common, is it just wetland plants or is it more than that? I can't remember now.
Bob's focusing on wetland plants for now, but he's trying to kind of expand what it will do over time.
Yeah, and so it's really fun. And the nice thing is you could go, well, especially if you're from West Michigan, you could go to those parks and after looking at what he put on the website, a lot of them are common plants that we as wetland delineators are familiar with, but not everything is. So yeah, especially like for me, the aquatics I don't know as well. So that was interesting for me to check out that part of it. So, okay, well, I'll shut this down. This was recorded. Unfortunately, I forgot to start recording it at the very beginning, but I did start the recording about 5 minutes into it. So that recording will be made available, I believe, on the MWA website. So anyway, thanks a lot again, Gary, and have a good weekend, everybody. And have a good rest of your field season. Bye-bye.